MHC2 molecules are involved in elimination of extracellular infections and generation of antibodies because MHC2 molecules bind peptides which have derived from outside the cell and have been processed by the antigen presenting cell in their endosomes and the lysosomes. So basically they process antigens which have an exogenous pathway. So whenever MHC2 cells bind to the T helper cells or CD4 T cells, they help activate macrophages and B lymphocytes to generate antibodies. Interferon gamma is one of the key cytokines which stimulates MHC2 expression on antigen presenting cells, that is dendritic cells and macrophages. B cells also have a constitutive expression of MHC2 on their surface, which increases upon antigen recognition and cytokine produced by T helper cells. Now let us compare the features of class 1 and class 2 MHC molecule. The class 1 MHC molecule is composed of alpha chain which has three domains, that is alpha 1, alpha 2 and alpha 3, and a beta 2 microglobulin. The alpha chain is quite larger as compared to the B2 microglobulin. MHC2 molecule is composed of two chains, that is alpha and beta, which are almost equal in size. So each chain has two domains, that is alpha 1 and alpha 2, beta 1 and beta 2. The peptide binding cleft in MHC1 molecule is composed of alpha 1 domain and alpha 2 domain. That is, these domains are the one which contains polymorphic residues. On the other hand, class 2 molecule has a binding cleft made up of alpha 1 and beta 1. So in this case, these domains have polymorphic residues. The alpha 3 region of the MHC1 class interacts with the T cell receptors of CD8 cells. On the other hand, the beta 2 region of MHC class 2 interact with CD4 T cells. The binding cleft of class 1 molecule can accommodate about 8 to 11 residues of peptide, so it is comparatively smaller as compared to class 2, which can accommodate a peptide about 10 to 30 residues. So in human, there are HLA A, B and C types of MHC1 class molecule, and class 3 molecules can be HLA-DR, HLA-DQ, HLA-DP.
The main antigen presenting cells are the B cell, the dendritic cell, and the macrophage. These cells can present antigen to CD4 positive cells. Additionally, all nucleated cells can present to CD8 positive T cells. All cells of the body express class 1 molecules encoded within the major histocompatibility complex, or MHC. Additionally, the B cell, the dendritic cell, and the macrophage express MHC class 2 molecules. These membrane molecules may contain degraded antigen peptides in a cleft on the top of the molecule, which they express to the T cells. The molecules regulate the way that the T cell reacts, since a T cell will only recognize foreign antigens when they are associated with the MHC molecules themselves. There are many varieties of class 1 and class 2 molecules, although in any one individual there may be up to 6 of each type. The Mhc gene locus contains 3 class 2 genes, DP, DQ, and DR, each formed by 2 chains, beta and alpha. It also contains 3 class 1 genes, B, C, and A. Because each individual has 2 chromosomes carrying these genes, the maternal chromosome and the paternal chromosome, There are usually 6 different versions of the class 1 molecule and 6 different versions of the class 2 molecule, produced from the 3 different genes with the 2 chromosomes encoding them. Each of the class 2 molecules is formed from an alpha and a beta chain. The indent formed on the top of the molecule is where the antigenic peptide is held. Because the MHC is dependent on the genetic makeup of the individual, it is very unlikely that 2 unrelated people will have the same MHC molecules. Each of the class 1 genes produces a single alpha chain which folds itself up and interacts with another molecule called beta-2 microglobulin, which comes from a different gene locus. Since each individual has different MHC molecules, the performance of everyone's immune system is different. This affects susceptibility to diseases where the immune system is.
A cell infected by a virus may degrade viral proteins. Class I MHC proteins bind bits of viral protein and transport them to the cell's surface for display. A cell displaying such foreign antigens on its MHC proteins is called an antigen-presenting cell. A cytotoxic T cell, or killer T cell, can bind the displayed antigen if the T cell has surface T cell receptors that correspond to a specific antigenic determinant on the displayed antigen. Once the T cell has docked, the antigen-presenting cell releases cytokines, chemical signals that activate the T cell. The activated T cell produces another cytokine and receptors for it. This cytokine causes the T cell to divide, rapidly producing a clone of cells that are identical to the first T cell in genetic makeup and immunologic specificity. Production of this clone completes the activation phase of the cellular immune response. The cloned T cells circulate in the blood and lymph. In the effector phase of the cellular immune response, ever they encounter them. The infected cells display viral antigen on their surfaces. When an activated cytotoxic T cell binds specifically to the displayed antigen, the T cell releases a protein called perforin, which perforates the plasma membrane of the infected cell, allowing water to enter and causing it to lyse.